Now inheritance of two genes. Inheritance of two genes, when we use two traits the same type, then it is called dihybrid cross. Three traits, then it is called trihybrid cross. Four traits, tetrahybrid. Five traits, pentahybrid. Many traits, polyhybrid crosses. Means number of pair of traits, contrast trait. Right? We have to use here hybrid plus monohybrid, single trait. Dihybrid, more than one, means two trait. Tri means three trait. Four means tetra trait. We can consider this one according to the number of contrasting traits we use here. Right? It is given inheritance of two genes. Before going to see the inheritance of two genes, other example of polyotropic gene is also here. As for example, in sickle cell anemia, it is also another. Sickle cell anemia means one phenotype is here. We understand that is means the RBC, they become sickle-like structure. That is the main characteristic of this one, this gene. Another also, it means forms the resistance against the malaria. Right? Sickle cell anemia means malaria. Malaria, that is our plasmodium. Plasmodium cannot survive in sickle cell like RBCs. So it gives also another one phenotype is a resistance against malaria. Right? Other thing also there means they are also responsible for the development of spleen, that is heart also, brain also, liver also here. Right? We can say. Okay. Now inheritance of two genes means dihybrid cross. Mendel also worked with the crossed pea plant that differ in two characters, as is seen in the cross between a pea plant that has seed with yellow color and round shape. So two traits are given here. One is yellow color and the other is round shape, and one that has seed of green color and wrinkle shape. Color and shape, two different traits, right? Mendel found that the seed resulting from the crossing of the parent had yellow colored round shaped seeds. In F1 generation, all the seeds have yellow colored and round shape, right? Here, can you tell which of the character is the pair yellow green color and round wrinkled shape was dominant? We can say it is easy to say because we know very well here, right? That is yellow and round, they are dominant, and simply second one is green and wrinkled. These two are recessive traits. The yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made the separate monohybrid crosses, right, between yellow and green. When monohybrid cross means when he took only one trait, that is yellow and green, so he get only yellow type of seeds in F1 generation. F2 generation means he get simply 1:2:1 ratio, is 1 yellow, 2 means it is also yellow, and 3 means 1, 3 means what? 1 means green. So phenotype means he get 3 yellow and 1 green, and genotype means 1 pure yellow, 2 hybrid yellow and 1 green. He get in case of monohybrid cross, right? Same as to here when he used round and wrinkled seeds. Round 3:1, round is 3 and 1 wrinkled phenotype. Genotype means 1 pure round, 2 hybrid round and 1 wrinkled, right? But let us use the genotypic symbol Y for dominant yellow seed color, Y for recessive green seed color, R round shape and R for wrinkled seed shape. The genotype of the parent can then be written as, genotype means it is the capital dominant traits and it is for the recessive traits. Now we have two types of gametes here. The cross between the two plants can be written down because we have two types of gametes here. Capital R and capital Y is there, and in case of this one, small R and small y. Two types only gametes are there. When they are crossed with each other in F1 generation, we get only what? Capital R, small r, capital Y, small hybrids. In F1 generation, we get hybrid. All the plants, they have only round and yellow seeds. The cross between the two plants can be written down here, showing the genotype of the parent plant. Parent which type? Dominant parent type, right? The gametes RY, as we write here, and small ry unite to fertilization to produce the F1 hybrid, that is capital R, small r, capital Y, small y, right? When Mendel self-hybridized the F1 plants, he found that 3:4 of F2 plants have yellow seed and one of the fourth had green seeds. Also, the yellow and green color segregates in 3:1 ratio, right? Round and wrinkled seed shape also segregate in 3:1 ratio, just like in monohybrid cross. In dihybrid cross also they segregate this, means they follow this one, right? Okay, we will discuss this one right after. Now he get the law of independent assortment by dihybrid cross, right? Why he get law of independent assortment through dihybrid cross? Because he get 4 types of seeds. Initially 2 types of parent seeds, from 2 types of parent seeds, right? He get 4 types of seeds, offspring, 4 types of offspring, right? In the ratio of 9:3:3:1. Okay, you can see here. That is round and yellow, and that one is another gamete. This one is green and wrinkled. When we cross this one, so this is the first generation, and all the plants, F1 generation, all the plants are round and yellow, right? Now selfing, right? After selfing, what happens? We get the ratio 9:3:3:2 when 1 here. This is a Pune chart, right? This side, these are gametes. 4 types of gametes are produced in hybrid cross. That is, it may their probability, their possibility. Capital R, capital Y, capital I. This is small r, capital Y, capital I, small y, and small r, small y. This side also same is there. Now this ry, it means capital R, capital R, capital Y, capital Y. This time small r, capital Y, small r, capital Y. We can write here small r, capital Y, capital Y. Here capital I, small capital R. Look here. This is capital R, capital Y, small r, small y. We can write this one, right? So you have to write this one only, which is you match this one. That side ry, that side ry. This one is. You can see here capital R, small y. That side capital R, small y. When you match this one, this is the capital R, capital small y, small y, right? So when we consider this one, you can see here 1, 2, 3, 4, 5, 6, 7, 8, and 9. 9 are yellow and round. Look here, 1, 2, 3. 3 are yellow and wrinkled. Here 1, 2, 3. 3 are round and green, and only 1 it is wrinkled and green. This is the phenotype, right? Phenotype. Easily you can understand phenotype and how many phenotypes are there. How? Now question, why this one? Suppose that we get this one. Why this one is? Only see, if one capital letter is there, that means one dominant character is present. Capital letter means capital letter, it shows the dominant character. So one capital letter present means this dominant trait is present in the offspring. Here capital R, capital Y present means round and yellow. Capital R, one capital Y is also there, round and yellow. Here only small r is there, that's why it is not round, it is wrinkled. But capital Y is there, that's why it is yellow. You can see here only small y is there, that's why it is green in color, no yellow color. But capital R present means round. In this case, you can see capital R, capital Y both are present, that is round and yellow is present. Do you understand? Here only small is there, wrinkled and green is there. Do you understand? If one capital letter is present, that means this trait is shows. Capital R present means round and capital Y present means yellow. Okay. If both are recessive means small r, both are small r, that means it is wrinkled. Both are small y is there, that means it is green. So you can understand this is a phenotype. And genotype, when we consider genotype, how could you understand which are the genotype? Simply you have to see, like here what is given. It is capital R, capital R, capital Y, capital Y. So we can write here capital Y. How many? 1. Second, look here, these two, right? Here, because we are seeing the genotype. Here, capital R, one R is small, one R is small. Here, 2, right? I write here capital R, small r, capital Y, 2, right? Now here, you can see here, capital R, capital R, capital Y, capital R, capital R, capital Y, and Y, capital R, capital R, capital Y, and small y. How many? 1 and 2. I should write here 2, right? And here, one is capital R, capital R, capital Y, capital Y. How many? 1. Okay. Now see this intermediate. R R Y Y, R R Y Y, R R Y Y, R R Y Y. I write this. These are 4, right? This one is capital R, small r, capital Y, small y. How many are there? 4. You understand, right? Now see this one. Small r, capital Y, small r, capital Y, small y. How many? 2 are there. 1 and 2, right? Here what is there? Capital R, small y, small y. How many are there? Only 1. Here, this one, now this line, right? It contains capital R, small y, small y. How many? 2. And this last one is small r, small y, 1. Total, when you count this one, we have 16. So what is the ratio? Simply, right, it is 1:2:2:1:4:2 1:2:1. Okay, you can, okay, I know you get forward. So you can understand here how many times 1 is complete. 1, 2, 3, 4. 4 times 1, 4 times 2, and 4. This is the ratio. 1, 1, 1, 1, 2, 2, 2, 2, and 4. 4 times 1, 4 times 2, and 4. That is the ratio. That is the genotype ratio. So we get genotype ratio through this square also. This one, now this and this one is there. It is the different. This one and this one is same. This one is different, right? You understand this one? How could we get the genotype and the phenotype? What is the genotype? How many genotypes are there? We know 1, 2, 3, 4, 5, 6, 7, 8, 9 types of genotype. Here we use one rule because for means when we want the phenotype. Phenotype means what? 2n, and genotype means what? 3n, right? And how many gametes if we want? That is 4n, right? So 2n means what? Phenotype. Here we know dihybrid cross. Dihybrid cross means what? 2 traits are there. N means what? Trait. How many traits we get? So because 2 square dihybrid cross means 4 phenotype, right? And 3n means what? Genotype. We understand here. 3n means what? 2. So you can write here 3 square means 3 multiplied by 3. How many? 9 genotype we get. And you can see here how many genotypes are there? 9 genotypes are there. Suppose it is tetramerous or pentamerous. Suppose that it is trimerous. Then what you understand? Trim is 2 multiplied by 2 multiplied by 2. 8 phenotype is there. And if it is means number of genotype, so simply we have to write this one 3:3:3 means 27 genotype, right? Do you understand this one, right? So through this one 2n or 3n, through 2n and 3n we can get the phenotype or genotype of a given hybrid cross. Do you understand? Okay. Now come how this law of how this dihybrid cross explain the law of independent assortment. Independent assortment means what? They are freely moved to anywhere else. Look, sometime what happens? This green color moves to the round shape sheet, right? And sometimes it is moved toward the wrinkle shape, wrinkle shaped shape. Means this trait moves freely, right? Same happens also for color. That is our for color means for shape. Wrinkle shape move toward the round seed. Wrinkle shape toward the green seed. Means they are independently move from one place to another place. We can say here through crossing over or other things, they move independently. That is called independent assortment, right? You can see here the phenotype round yellow, round green, and wrinkled green appear in the ratio of 9:3:1. 9:3:1. Such a ratio was observed for several pairs of characters that Mendel studies. The ratio 9:3:1 can be derived as the combination series of 3 yellow, 1 green in case of color, 3 dominant yellow and 1 green phenotype, 3 round and 1 wrinkled. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green. This is the ratio 3:1, 3:1 in both traits, right? That is 9 round. When we multiply this one, 3 round multiply 3 yellow, that is what? 9 round and yellow, 3 multiply by 1, so 3 round green, that is 3 round and green, right? Okay, it is given green and yellow and wrinkled. When we multiply this one, 3 multiply by this one, right? 3 round green, 1 wrinkled green. So you understand how it gets? You can see here, when we multiply by 3 into 3, this 3 into 3, right? So we get 3 into 3. Now we get 9 round and yellow, right? When we multiply this 3 into 1, so we get 3 round and green. Do you understand? When we multiply this 1 with 3, so we get 3 yellow wrinkled capital Y and small r, right? And when we multiply this 1 into 1, then we get 1 wrinkled and green small r small r. That is given. You understand how it, right? Based upon such observation on dihybrid process, cross between plant differing in two traits, Mendel proposed a second set of generalization that we call the Mendel's law of independent assortment. Law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. It is totally independent. They are not interrelated. They are not interdependent. They are free, right? So they freely move. That is given. The Punnett square can be effectively used to understand the independent segregation of two pairs of genes during the meiosis. This Punnett means it is a scientist who used this Punnett square, right? This one is a scientist who used the square to describe the possibilities. Here, why it is given? The Punnett square during the meiosis. What is given here? Punnett square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of eggs and pollen grain in the F1 generation, that is hybrid plants are there, capital R, small r, capital Y, small r, right? Only dominant character, only dominant trait is expressed here, but it is hybrid, we know. Consider the segregation of one pair of gene R and R. 50% of the gametes have the gene capital R and other 50% they have gene small r, because it is hybrid, we know. Now, beside each gametes having either R, they may be either R, capital R or small r. It should always have allele same as to capital Y or small y. The important thing to remember here is that segregation of 50% R and 50% small r, it is independent from the segregation of 50% of y and 50% of small y. They are independent. They can move anywhere else, right? Therefore, 50% of R bearing gametes has Y and other 50% has small y, right? Similarly, 50% of capital R, they bearing gametes which have capital Y and 50% they have small y because they can move independently. Thus, there are 4 genotypes of gametes. We get capital R, capital Y, capital R, small y. Then second one is small r, capital Y, small r, small y, right? 4 types of pollen and 4 types of eggs are produced. The 4 types are capital R, capital Y, capital R, small r, capital Y, and small r, small y, each with the frequency of 25% of 1/4 of the total gamete produced. We can say there may be 4 types of gametes, means 20, 25, 25, 25, 25 possibilities there, right? When you write down 4 types of eggs and pollen on 2 sides of the Punnett square, it is very easy to derive the composition of the zygote that give rise to F2 zygote. Number of zygote, it is obtained by 4n. Here, I already tell you here, for phenotype, we use 2n. For genotype, we use 3n. And for zygote, when we need zygote, how many types zygotes are there? We use 4n. Right? In a dihybrid cross, we know how many phenotypes are there? 4 phenotypes are there. Genotypes means how many? 9. And zygote means how many? 16. Simply because this n, it indicates the number of traits. And in case of dihybrid, it is 2. So these are the right number of offsprings we get here. Here, although there are 16 squares, how many different type of genotypes and phenotypes are formed? Already I tell you how many different types of phenotype, 4 types. And what is the ratio? It is 9:3:3:1. And how many types of genotype are there? We have 9 types of genotypes, and the ratio is 1:1:1. 2:2:2:2 and 4. You understand? 4 times 1, 4 times 2 and 4. That is the ratio. Note them and down in the format given. Right? Note them down. Okay. Now, can using the Punnett square data work out the genotype ratio in F2 generation and fill the format given below? Is the genotype ratio is also 9:3:3:1? No. Right? Already we discussed here. We need not to once more discuss. Right? Genotype found in case of dihybrid cross. In case, suppose that it is a dihybrid cross. So dihybrid means 2 traits we use. The genotype means F2 generation, it is 4 genotypes. Right? But when we consider the phenotype, no, it is genotype. Genotype means what? 9 genotypes are there. And expected phenotypes are 4 phenotypes. Right? But when it is trihybrid cross, then how many genotypes are found? Genotypes. Genotype means 3n. So 3n means how many genotypes are there? Then it is 27 genotypes. And expected phenotypes means how many phenotypes are there? 8 phenotypes. Right? Tetrahybrid. Tetrahybrid means we have to multiply this simply, and we can understand how many genotypes are found and how many phenotypes are found here. Right? When it is a trihybrid cross, the expected genotype is too much, right? Ratio of genotype is too much. But we can expect the phenotype means 27:9:9 is because how many? Here 8. 8 means 27:9:9:9:3:3. We can get here 29, 27, right? This 27, right? 27, 9, 9, 9 is 2. 3 is to 3 is to 3 is to 1. That is the phenotypic ratio in case of trihybrid cross. Dihybrid cross, we know here 9 is to 3 is to 3 is to 1. That is the ratio, right? Now, next point is chromosomal theory of inheritance. Before going to chromosomal theory of inheritance, first I tell you, suppose that a polygenic inheritance. In polygenic inheritance, what happens? Polygenic inheritance means what? When more than one gene is responsible for express a specific trait, a single trait, then it is called a polygenic inheritance. You can see here. It is also present in our syllabus. So I write here polygenic inheritance. It means what? One character is controlled by more than two genes. We can say more genes. Means 3 genes, 4 genes, 5 genes, right? More than one gene. Simply you understand more than one gene. Then it is called as polygenic inheritance. As for example, it is also present in case of plant. Plant means color of wheat kernels. Which is controlled by two genes or two alleles, right? Another is all that is red and white kernels. As for example, red and white kernels, right? And second is our skin color of human. Skin color of human. It is regulated by 3 genes. 3 genes, right? This one this one is 2 genes means which one? R and B. Or small r, small b, small b, right? And it is controlled by 3 genes, and these genes are capital A, capital A, capital B, capital C, and small a, small b, small c, small c, right? So you can understand here, suppose that in a color of wheat kernel, it is controlled by 2 genes, and we understand here we have two types of gametes are there. Either they are, one is capital R, capital B, another is small r, small b. In case of F1 generation, all these wheat kernels, when we consider this one, means first case, what happens? We have two types of gametes are there. When it is crossed with capital R, capital B, crossed with small r, small b. In F1 generation, we get capital R, capital B, small b. Means here all these are they are intermediate. They are not, they are intermediate, right? We cannot say these are red. This one is red, and this one is white, right? Only one trait is there, because one trait is controlled by more than one genes, that is 2 genes, right? We get intermediate color in F1 generation. This one is F1 generation, right? And in F2 generation, what happens? After means we can say selfing. F2 generation means what? Now we have how many? We have here, because here it is depend upon the number of dominant traits, right? We have different types of color of kernels are present here. So here we understand, because we have which one is there? F2 generation means different. Different means what? Here 4 these are there. If 1, 2, 3, 4, all these are all capitals are present. If all capitals are present, then it is totally full red. We can get here capital R, capital V. Second means capital R, capital V, small v. Third means what? Capital R, small r, capital V, small v. Fourth means what? It may be small. Here what happens? 1, 2, 3, 4 means 4 they are dominant. Here 3 are dominant. Here 2 are dominant. And in this case what happens? Only one is dominant. Capital R, small v. Only one is dominant, right? And here what happens? Small r, small v. No one is dominant, right? No one is dominant. So that means what happened here? Zero dominant. You can write here zero dominant characters are there. So zero dominant character means what? White. Here we can say it is white. This one is color of white because small are there. This one is means we cannot say this one is a red or white. This is light. You can say very light color. Very light, near about to white, right? Here it is intermediate. Intermediate means simply we can write intermediate, right? This one is light red and this one is red color, right? Now what is the ratio? When we consider ratio, then what is the ratio? In this ratio we can understand to polygenic inheritance, right? Ratio means what is the ratio here? Ratio we get here 1, right? 1, this is RRBB, right? Full red means dominant gene is 4. 1 is to 4 is to 1 is to 4 is to 6 is to 4 and then 1. You can write here ratio is 1 because only 1 is there. 3 capital 1 dark. It is how many? How many are 3 capital and 1 dark? 1 dark. When you sign the Punnett square, then you understand, right? Here we get how many? It is 6. Sorry, 4. 4. How many? Half means what? 6. How many these are? 4. And how many? 1. That is the phenotype ratio. This one is phenotype ratio. 1 is to 4 is to 6 is to 4 is to 1. Right? One thing is there, in case of polygenic inheritance, number of dominant gene is responsible for the specific trait, for the efficiency of specific trait. It is present in case of red and white, weak means carnal white. Okay, second is our given skin color of human, because skin color of human is present in our NCERT. So we have to see how a skin color of right. Skin color, as I already tell you, skin color it is controlled by 3 alleles: capital A, capital B, capital C, or small a, small b, small c. Right? Because it is a tri-hybrid cross, we understand tri-hybrid cross means how many how many zygotes are formed? 64 zygotes are tri-hybrid cross. Because 3 genes, 3 alleles are there. You can see here how many we have in case of skin color of humans. We need not to draw here. Okay, when you have free time you can draw, right? Because 68 squares are formed and it is too much, right? So how many? It is capital A, capital A, capital B, capital B, capital C crossed with small a, small b, small b, and small c, small c. So this one is Negro. We can say Negro, black color is there, and this one is it is not a Negro. We can say it is a melanism, or we can say here white color, right? This fair skin, white color skin. When it is crossed in F1 generation, this one is F1 generation. In F1 generation, we get intermediate. Intermediate. These are intermediate. Means we can get here capital A, small a, capital B, small b, capital C, small c. Right? In case of submitting time, then what happens? This one is F1 generation. So these are called mulatto. This color is called mulatto color. Right? Mulatto. So on selfing, we get different, means intermediates are there, right? And here we can get what ratio we get here? Here we can say 1, 6, 15, 20, then after 15, 20, then after 15, 6, 1, right? We get the ratio here 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. That is the phenotype ratio. Of different color of skins, right? And you understand how I write this one, because it is this. How many? We have 6, 1, 2, 3, 4, 5, 6. 6 genes are there. In 6, I write here also 6, 1, 2, 3, 4, 5, 6, right? 1, 2, 3, 4, 5, 6, 7. Okay, 7 is given, right? So how I write here? Look here. Here all are dominants. All are dominant, right? Means here how many dominant? Here I write dominant traits. Number of dominant traits. So when we consider the number of dominant traits, here how many? 6 dominant traits are there. But here 5 dominant traits. Here 4 dominant traits. Here 3 dominant traits. Then after here also 4 dominant traits. Here also 3, not 4. It is because we are going in reverse order. Here 2 dominant traits. Here 1 dominant traits, and here 0 dominant traits. So this side, this one is, it is not a ratio, right? We can say here, right? It is not a ratio. This is the number of dominant traits, right? Number of dominant traits. 6 dominant traits means what? We can write here F. Capital A, capital A, capital B, capital B, capital C, capital C. In this case, you decrease one by one. Capital A, capital A, capital B, capital B, capital C, small c. Small c is there. Now here 4. 4 means what? Capital A, capital A, capital B, small c and small c. These two are small, right? These two are small. Simply we can say. Right, here we need write put like this one. Capital B, small b, capital C, small c. This one is also capital A, capital B, small b, capital C, small c. 3 capital, 3 small. Here also 2, 1 is only 2 is there. So 2 means what? Capital A, small capital B, small b. Small c, this side capital A, small b, small c, and this side small a, small b, small c. The number of dominant traits is responsible for the coloration in the skin. That means all the dominants are there. That is, this one is, it is called, we can say it is totally black negro, right? This one is, look, black negro. This one is some light negro. We can say this is very brown. Look, not a black, but it is very dark brown. This one dark brown, right? And this one is mulatto, intermediate. So we can say mulatto, 20 number of. And this one is, look here, dark brown. Now here start light brown. This one is very light brown. and this one is white one. So these are the different color which is present in human, and that is how they produce. Because here polygenic inheritance is there. In case of polygenic inheritance, number of dominant traits are responsible for the expression of specific trait. Okay. So today we finish our lecture here, and next day, I will tell you about the we can say that is the next portion is our chromosomal theory of inheritance. Right? We will discuss about this chromosomal theory of inheritance in our next class. Okay? Thanks.